Welcome everyone to the third ReSVINET webinar. My name is Bont. I'm the chairman of the ReSVINET Foundation, and I'm very happy to see that so many of you have joined us today for the webinar. I will do a short introduction what the foundation is about. Our vision is to connect global expertise and leadership in order to decrease the global burden of RSV infection, with a mission to support performance of high-quality research, to improve knowledge of RSV epidemiology, and to develop safe and effective therapeutic and preventive interventions. With the specific goals to advocate and create awareness, to act as a focal point for effective partnership, and to combine knowledge and capacity required to enhance development of novel therapeutics. So why do we actually do what we are doing? Why RSV research and education? And I think you're all aware that there are so many patients, 3 million hospitalizations, at least 120,000 deaths, most of them in the first 5 months of life. Good enough reason to continue doing what we're doing. So for today, we have the objective of the webinar series to promote knowledge of the latest developments and publications on groundbreaking studies, and to provide education to young professionals in the field of RSV with a frequency of 6 webinars per year. Today, we will hear about neutrophilic inflammation in the respiratory mucosa as a predisposition to RSV infection, with Peter Openshaw, who will be shortly introduced by Linde Meyaard. She's an immunobiologist here at the University Medical Center in Utrecht, where I work. She is my direct colleague. She has been that for more than 10 years now, and we join a translational research team where we do all our studies, and with a specific interest in neutrophil biology in RSV pathogenesis. And I just wanted to highlight that March 25, you can mark that in your calendars, you will get the invite anyway, that we're going to hear about the impact of COVID on seasonal viruses and obviously on RSV epidemiology, in particular by Dan Weinberger from Yale. Without further ado, I'm now going to hand over the microphone to Linde Meyaard, who will introduce Peter Openshaw. Linde, the floor is yours.
Thank you, Louis. It's my great pleasure to introduce Professor Peter Openshaw, who is a respiratory physician and a mucosal immunologist who's been working on respiratory infections, as we just learned, since the mid-80s. And he has a long-standing career studying the pathogenesis of this type of respiratory infections. And in this function has chaired many consortia that are focused on understanding the pathology of respiratory infections. And I think the main reason he is here right now is that he directs studies on human volunteers to be experimentally infected with this type of viruses to really understand what's going on. And I think with this, we can learn so much more than we can learn from animal models. So it is with great interest that we are looking forward to your talk. And recently, of course, all these studies on respiratory infections has been gaining increasing interest because of the COVID pandemic. And it's my understanding, Professor Openshaw, that also in this field you are active. So I'm looking forward to your talk, and I look forward to enjoy the question and answer session after you are finished. The floor is yours.
Well, thank you so much, Linde. It's a great pleasure to meet you and to see old friends, Louis Bont. I see my old friend Jesse Grottas is online as well. So, you know, it's a great pleasure to be here to focus a bit on the neutrophilic aspects of inflammation due to RSV infection. Obviously, in this forum, we don't really need to introduce the virus much, but I thought I'd just show this picture in case anyone hasn't seen it. I think it's a lovely depiction of RSV structure. We when I was doing my PhD many years ago, I spent a happy afternoon in the EM suite looking for a typical bit of RSV in a preparation, and never managed to really find anything that looked like what we thought the virus looked like at that time. And actually, it seems that the infectious part of the virus is filamentous. In terms of the disease it causes, this is a picture which we've recently put into a review which came out in Thorax just at the end of 2009, which I think illustrates the type of disease that is caused by the virus. You can see in the middle a bronchiole, which ought to be full of air, but it actually is in fact full of inflammatory cells. And a lot of these cells are in fact neutrophils. So if you wash out the lungs of children with bronchiolitis and obtain cells from the lungs, neutrophils are a pretty predominant type. So the question is, what are those neutrophils doing there? When I first saw this in the mouse model, the neutrophilia that we sometimes can see under some circumstances, I thought maybe I'd got a bacterial contamination and that this was really a bacterial secondary infection. But I think it's now clear that the neutrophilic response is just a response to the actual virus itself. Now, this is some data which is also in that review in Thorax, which is in fact from a Dutch study where the authors looked at the frequency of respiratory disease in people of different ages in the whole of the Netherlands. And they divided this into people who were young, age 0 to 4, and old, over 75. And they looked at the epidemiology during the period of the 2009 pandemic. And they didn't find a huge increase in the frequency of admission or death due to influenza. But incidentally, the control group, the over 75s, they did see an association between the circulation of RSV and deaths in older adults. And the reason I wanted to highlight this is that RSV is not only a pediatric disease looked after on the wards of Louis Bont, but is also apparent in the older adults. And if you look at this same data set from the Netherlands in terms of the number of deaths which are projected to be due to RSV in older adults and in young children, the deaths amongst older adults are in vast excess compared to the younger children. So I'm just trying to make the case here that RSV is not just a pediatric disease, but is an important disease, often with hidden effects in older adults. This is a diagram which comes from a review that we wrote a couple of years ago and illustrates the RSV effects at different ages. Obviously in young children it causes bronchiolitis, but then it has this association with recurrent wheeze in later childhood, which Louis has brought to the attention of the world in a very dramatic ways. And then in older adults, something which Anne Falsey and others have been working on for a long time, RSV can present in ways which are sometimes hard to diagnose. The typical story might be that grandchildren come home at Christmas, they have slightly runny noses, maybe they're recovering from RSV. They pass it on to the grandparents who may be suffering from chronic obstructive lung disease. And over the next couple of weeks, they get a cold, and then the cold goes to their chest, and then they deteriorate, and they might be admitted maybe 3 weeks later, by which time it's very hard to actually discover that it was RSV that triggered that deterioration of their chronic lung disease. So it is a disease of all ages. So we've been looking at the possibility of infecting adults and indeed older adults with RSV experimentally in order to investigate the pathogenesis in more detail. And just in outline, we found that if we expose people to RSV by intranasal drops, then a proportion become infected, but the proportion remains uninfected. And those who become infected, only a subset actually develop symptoms. So it's very interesting trying to understand what it is about the different volunteers that explains that variability. So you might think that experimental infection was something that was done back in the 30s, 40s, 50s, but actually it's died out. In fact, the opposite is true. Experimental infection of human volunteers is in the ascendant. There are more infection studies being done in volunteers now, and it's something that, again, we in the UK have in common with our friends in the Netherlands, which is that we are both very active in this field. There are quite a few investigators who are performing this type of experimental infections on both sides of the channel. I actually lead a consortium funded by the Medical Research Council, which is called HICVAC, which is the Human Infection Challenge consortium to accelerate vaccination. The purpose of this consortium is to try to support work which will accelerate vaccine development. At the moment, we are actually considering doing human experimental challenge with COVID-19. It's a difficult area, but I think it could be very revealing if we do go ahead with that. So in terms of the human volunteer studies, this is Max Habibi, one of our research fellows who's demonstrating the method of inoculation, which is very simple. We just drop virus into the nose. It's this Memphis 37, which John De Vincenzo initially isolated from a child with bronchiolitis. We keep the volunteers in seclusion, not in total isolation, but in seclusion in a research facility, day and night for a period of 10 days. And that's largely because we're taking an awful lot of samples from them, and we're often doing repeated bronchoscopies. And it's just more convenient, really, to keep them in a facility. I think it would be possible, theoretically, to do this in an outpatient environment. So we've discovered quite a lot of interesting things about the infection in adults by doing this. One is, I think, a bit surprising, which is that the symptoms resolve almost completely, but we still get quite a lot of airway inflammation, which you can see at bronchoscopy. And this is a day 28 bronchoscopy on an individual who had actually recovered completely, had no symptoms at all. But actually, you can see not only airway changes, but also some evidence of viral antigen in the airways at that stage. So you remember that I said that that not all volunteers behave the same. So we give them all virus. And then in this particular study, out of 58 volunteers, 23 developed a cold and 25 had no cold. So we've just divided them simply into those two groups, quite nicely balanced groups, which we can then study and try and work out what's different between those who do get colds and those who don't. So in those who do get symptoms, there's a lag, a time delay between the inoculation of the virus and the appearance of symptoms, and that is roughly 3 days. So it's only on day 4 that symptoms begin to develop using the virus that we're inoculating. So this is what we call the incubation phase, and then we get viral replication, and the virus peaks at about the same time that symptoms peak. So what makes the difference between somebody who gets a cold and somebody who doesn't? Well, you might expect that it would be antibody. So this is the level of anti-RSV IgA in the nasal secretions, and you can see that there is a little bit of a difference statistically between those who get a cold and those who don't, but it's by no means, sorry, I'm just needing to adjust my slides, but it's by no means the determinant of whether somebody gets infected or not. It's only a trend. So something else which we have looked at, because we're doing bronchoscopy, we're able to specifically sample the airways and look for T cells in the airways, and therefore to demonstrate whether there's any airway resident memory CD4 or CD8 cells that may predict infection. And indeed, it does seem that the presence of these resident memory CD8 cells may have an effect in terms of the recovery from viral infection. And we think that that may be because of CXCL10. There's a very tight correlation between CXCL10 and the recovery from infection in this particular model. And that, I think, demonstrates what T cells can do. They're obviously not doing an equivalent thing to what antibodies do. And obviously CD4 cells are important in terms of sustaining the antibody response. Okay, so it's a great model, and it's allowed us to go back and try and work out what was it about the state of the respiratory mucosa that predicted the outcome. And we can do this using a device called a rhinoprobe to scrape mucosal tissue out of the inferior turbinate prior to inoculation of the virus. So that gives us a baseline estimate of the transcriptomic signature present within the airway cells before the virus goes in. So this is the sort of thing which you could never do by an opportunistic study where you observe people with infection. You couldn't practically sample the tissue in the nose and then go ahead and inoculate or watch the effect of natural infection. So this is a great advantage of the controlled human infection system. So when we look at the transcriptomic signature in the nose, we do indeed get a very big difference between those who have a cold in the upper part of the panel and those who don't have a cold in the lower part. And when we look at the genes that are associated with this, the genes that most tightly predict susceptibility are genes indicating neutrophil activation, neutrophil response, neutrophil immunity, neutrophil degranulation. So all the top hits in this type of screen are to do with neutrophils. So elevated neutrophil activation at baseline is the factor that distinguishes those who go on to get a cold. We can also use another technique called nasabsorption, the SAM strips, which were developed by Trevor Hansel, whose very fine nose you can see here demonstrating his SAM strips. So Trevor and I have worked together with great pleasure for about 10 years. He unfortunately is just retired, but I think this was one of his greatest contributions to the study of the mucosa, is to develop these beautiful strips which are now available commercially. And if any of you want to watch a video, there's a nice video in the Journal of Visualized Experiments, I think it's called. And that's nice to watch. Anyway, so Ryan Thwaites did some sampling of the nasal mucosal fluid using these strips and showed that we could demonstrate various factors that are related to the neutrophilia, neutrophil products or cytokines, chemokines that trigger neutrophils are present abundantly in those who develop colds. So really corroborating that signature which we see on the RNA. So an obvious question is whether this neutrophil signature, which makes people susceptible to getting RSV infection, is driven by the bacterial flora in the nose. And we did a pretty hard search for this. We thought it was going to be the explanation. We thought it would really clinch this study if only we could demonstrate that. But in fact, we were unable to. There was no clear signature in amongst the bacterial screening that we did based on 16S that told us that there was any particular bacterial species that determined the neutrophil excess in those who went on to develop colds. But I would say that the study was maybe too small to look for those differences, given how diverse the bacterial flora is. Okay, so going back to the animal models, which I worked on so happily for many years to try to look at the mechanisms of this, Cecilia Johansson in the department was able to show that if we administered CXCL1 via the airway in mice, this draws neutrophils into the airway, and this also caused an enhancement of the severity of infection in mice as measured by weight loss. It didn't particularly affect the amount of virus that replicated in the latter part of disease. There was a bit of an effect in the early part, but it seemed to be that the disease was being modified by the chemoattraction of neutrophils into the airway by this maneuver. And she was further able to show that if you depleted the CD8 cells, which you can see here on this graph in green, if you deplete the CD8 cells, then that effect of chemoattracting in neutrophils is more or less ablated. And I really like these experiments because it took me right back to when I first started working in the RSV mouse model back in 1980s, mid 1980s. And we did studies where we injected CD8 cells in via the tail vein and then infected the mice intranasally. And the main thing that we found when we looked at the lungs of those mice was neutrophils. So again, there's this tight connection between the CD8 cells and neutrophils, which are almost a sort of readout of the attack on the infected lung that is committed by the CD8 T cells, which are cytolytic and cause lung damage. So that was what happens if you look at the pre-existing state of the mucosa and what happens if you alter the pre-existing state of the mucosa in the mouse. So what happens during this incubation phase? So we're now really concentrating on the first 3 days after inoculation of the virus, during which neither those with colds nor without colds have yet developed any symptoms. And here we've been using Trevor Hansel's wonderful SAM strips to measure what's in the mucosal fluid, and we've measured quite a large number of different chemokines and cytokines. And I'm not sure whether you can see this very clearly on the screen. For me, it's much too small, but I can tell you that this one at the bottom right is TNF. Then to the left, we've got IL-10, then we've got RANTES. So many of these different mediators actually decline in this incubation phase in those who go on to develop a cold, and then rebound during the phase when the cold is most exuberant. So if the mucosa goes into a quiescent stage during this incubation period, then the virus triumphs and a cold develops. Whereas if you look at the blue lines, these are people who didn't develop a cold, you can see a very slight increase in things like IP10, in interferon gamma, and maybe MIP1 alpha, particularly during this early incubation phase. And if the mucosa does mount a slight response, that's enough to defeat the virus. So that seems to correlate with the pre-existing determination, which is associated with this neutrophil activation signature. So there are a number of explanations for this. And again, I hope you can see this larger on your screen than I can on mine. There was a very nice accompanying editorial when we published this recently, which I think gives a nice explanation of the relationship between the airway neutrophilia and what's happening in the prodromal stage and the subsequent development of disease. In the middle panel, you can see that's illustrated from the accompanying editorial, which I think is a very, very neat way of explaining it. And on the right, there's a couple of cartoons from somebody on Twitter, which I really appreciated because I think they put the whole of the study in nice, simple diagrammatic context. And I often use that when I'm explaining this to students. So I think in summary, we know that primary RSV infection in babies is associated with neutrophilia. And we know that airway neutrophilia prior to exposure in the adult challenge model is associated with subsequent development of a common cold and of disease. And that there is this interesting association between CD8 cells, which I first observed many decades ago in the mouse model, and the inflammatory response which is seen. I think we still don't quite understand what it is that determines this susceptibility in terms of the changes in the neutrophils in the healthy volunteers. My hunch still is that there is something about the microbiome in the nose that may determine this, or maybe the recent infection history. But I think it is a really intriguing and potentially important finding. How could we manipulate that to our advantage in terms of being able to prevent colds? And we know that there are some conditions of inflammation in the lung and in the airways, which is associated with neutrophilia, and which can also be associated with a greater propensity to disease. So just like to finish by saying an absolutely huge thank you to the team that's been doing these challenges, and in particular to Chris Chu, who you can see in the pink shirt here, who is an infectious disease clinician who was my research fellow for several years and has now gone on to become a reader at Imperial and is part of the infectious disease team, and many others, but also Ryan Thwaites, who's been working with me very closely, and with Trevor Hansel on all this mucosal sampling. So I hope that's of interest to you, and thank you very much indeed for listening.
Thank you very much, Professor Openshaw. I think this clearly shows that you can get very unexpected findings by just make an inventory of what you see first, and then next try to interpret the findings. And there's a couple of interesting questions in the chat, and I have some questions of myself. I try to group them a little bit on topic. So the first set of questions comes from Victoria Jenkins and Luan Vu, who both ask if there was any correlation to susceptibility to the cold with systemic neutralizing antibodies against RSV.
Yes, so we did look at that. There is a very weak association, but in our study, it was less strongly associated to mucosal IgA. So it really was not predictive to any significant degree. And then I think what is, of course, a question you already raised, but I would like to get into a little bit more, is what explains the neutrophilia in these healthy individuals, and can we understand why it's there? So one of the questions comes from Mauricia Caballero, who asked, can this neutrophilia pattern be correlated to neutrophilic types of asthma? Yes, that's very interesting, isn't it? So, yeah, there's a lot of work which has gone on to look at those different types of asthma. And, when I was sort of growing up in respiratory medicine, it was very much an eosinophil-associated disease. But I think it's very clear that there are these different endotypes. We've done some other challenge studies using various TLR ligands, which we've squirted into the noses of volunteers. And in that case, using resiquimod, which is R848, which is something which is used to try to stimulate the immune response in people with warts. When we squirt that into the nose, if we give a high dose, it actually causes a systemic effect. People get a bit of a flu-like illness. If we give a low dose, then we get a nice, fairly predictable pattern of mediator release. And in people with a history of asthma or atopy, then that response is greatly enhanced. So I think, we'd like to investigate this further and try and work out whether there is some sort of baseline inflammation, which could be neutrophilic or in other types, that generally predict these type of nasal responses. But there's a lot of different work to be done here. I think.
Yeah, so I think what Stefania Cormier also relates to is there some sort of heterologous immunity? So does this just differ between people? So I was wondering, did you ever follow a healthy person over time and just see if having many neutrophils in your nose is just something that is, a trait of a person that doesn't change?
Sorry, we haven't done that, and it's such an obvious question. I would really like to have a research fellow do that prospectively and just look over time, not only at the signature which you get from the scrapes, but also at what we can measure using the SAM strips, and just do a basic study of what happens over time with exposure to dust and pollen and so on. It's quite a big undertaking in itself, and we've always meant to do it but haven't got around to it.
In addition to that, do you have any insight on how this neutrophilic phenotype would reflect children? Do you see the same there, this heterogeneity, or do you expect it? And do you expect it to play the same role in susceptibility as it does in adults?
Well, that would again be a great study to do. I don't know whether you could do nasal scrapes on children. We have managed to get some limited gene signatures out of the noses of babies just using SAM strips and extracting the RNA. So I think it would be possible in a prospective study to do that, and maybe you have such a study underway out of which you might be able to extract such a signal. It'd be very interesting.
I see Louise smiling. Louise, would you like to comment?
Thanks. Yes. So what we have been doing over the past years is trying to measure transcriptomics in cells from the lower airways of children with RSV bronchiolitis. And surprisingly, we were able to get those cells and really isolate neutrophils and get the transcriptomic profiles of those neutrophils. Actually, Shanna Westerman from our group did that. And we were absolutely sure that this is technically feasible in those who are mechanically ventilated. The challenge then is that you need to distinguish the RSV impact from the ventilator impact. So what we have been doing is try to get the samples as soon as possible, but those who are in the field of mechanical ventilation know that transcript changes in transcript profiles change really, really rapidly. That is still the challenge. We haven't gone as far as measuring neutrophil transcript profiles from the upper airways. So yes, this is something that I think you, Peter, and us are going to do in the upcoming promising study. Dig deeper in the mucosal neutrophil biomarkers, RNA biomarkers in the target population. Yeah.
Yeah, it'd be nice to be able to do a prospective study where you saved the nasal SAM strips and then analyze them once it's determined that you've got a group that goes on to have bronchiolitis or RSV infection and a group that doesn't. That would be really interesting.
I agree.
Yeah.
So I see that Laurine Rijsbergen asked exactly the same question about the difference between infants and elderly. So there's many people cheering for this study already in this group. She is also asking, together with Adrian Wildfire, about previous viral infectious exposure. So specifically, Adrian says, is it likely that prior RSV causes the transient up or down regulation in this incubation period? And could this be strain related? And Laurine has a similar question where she says, have you screened for viruses in the samples taken before inoculation, as it has been shown, for example, in influenza, that prior infection with rhinovirus leads to less severe influenza infection? So I guess both questions point to the same thing. Did you look at viral exposure, and do you think this is meaningful in this setting?
Yeah, I think it's one of the main, most likely explanations is that the mucosa, the state of the mucosa is affected by intercurrent common infections. We did screen all of our volunteers for other viruses and excluded anyone who was harboring any other virus. I think we had to remove one because they had rhinovirus present in the swab. But essentially, we were only taking people with no recent history of a cold and no demonstrable other infection present. But I think it's an obvious explanation is that some other infection history. Adrian asked about the strains of RSV. I'd love to repeat this whole study using different strains. That would be very interesting. And I think Adrian would probably predict that some of them won't have the same interval between inoculation and development of symptoms, but maybe dose related.
So then to allude a little bit still on the topic of the microbiome, Wouter de Steenhuizen comments that he read your paper with great interest and concluded that you did the microbiota analyses on throat instead of nasal samples. And do you think that this would make a difference in the outcome, and that the throat and nasal microbiome could be different? And could you comment why you chose to do it this way?
Yeah, well, we chose to do it that way because we could only really get a good signal at that time. I mean, you have to remember that a lot of this study was done almost, it's taken a long time to analyze all of this and to bring it to publication. I think if we were starting again at this stage, the way that we would analyze the microbiome might be more sophisticated, might be able to demonstrate more fine subdivisions in terms of the presence of different microbiota. And obviously, we would like to look at the microbiome not only in the throat, but also in the nose and potentially in the lung as well. But it's, we weren't able to investigate it any further with our particular samples, unfortunately.
I understand. So coming back to what these neutrophils do, maybe you can answer again 2 questions at the same time. So Peter Rigaud is asking what type of symptoms these volunteers experienced. And then Sofia Buonacore says, do you think those neutrophils play a role in disease severity? So maybe you could take those 2 and comment.
Yeah. So the main symptoms that people get are rhinorrhea. Occasionally they have a cough as well. The symptoms are generally pretty mild in this experimental model. And we didn't have anyone who had what we would call anything severe that lasted longer than about 24 hours. So generally pretty mild. I mean, we haven't really driven the model to the extent that we are trying to induce any more severe disease. I think that would be undesirable. The other part of the question was about...
Well, it was basically if you think those neutrophils play a role in disease severity, and I guess in this model there's no such thing as severe disease. But maybe I could add to that question and ask, at what level do you think these neutrophils stop the infection? Because do you think the virus doesn't even pass the nose, or do you think there's another? Another.
Yeah. So we can find the virus lower down in the respiratory tract. So it's not just confined to the upper respiratory tract, but we do get lower respiratory symptoms, but they tend to be fairly mild. As to the role of neutrophils, I'm not sure how much the neutrophils are just a signifier of disease and how much they're involved in the pathogenesis. That isn't perfectly clear to me. I mean, they're present in the airways of babies with bronchiolitis, but we're not quite sure in terms of function what they're doing there. So they're capable of degranulation and so on, but neutrophils have very diverse functions and also a very, the transcriptome of neutrophils is quite plastic in response to infection. And it may be that they are circulating back to bone marrow, for example, and have functions which are separate from the causality of disease.
Yeah, so to allude to that a little bit, it's known that neutrophils are not neutrophils. There's lots of neutrophil subsets described with different functions. So do you expect that these nasal neutrophils are of a different type than the ones you would find attracted to, for instance, a bacterial infection, or the ones that you find in bronchiolitis in abundance in the lower airways?
Yeah, I think for that we need to work with neutrophil biologists, and actually, that was a shortcoming in our study, which is that we didn't really expect to find this strong neutrophil signature, and we weren't really looking for neutrophils or trying to characterize them initially before we'd analyzed the transcriptomic signature and then gone back to look for other soluble markers. So I think if we were doing this prospectively, we would be wanting to work very closely with neutrophil biologists and, as you say, look at different subsets.
With a little bit alluding to that more, Sofia Buonocore asks, did GM-CSF came up in your study like what you described in COVID?
Yes. So not so much. Yeah, so the studies that we're doing on COVID, we do find that GM-CSF is one of the markers that shows a particular gradient in terms of association with disease severity. We're very intrigued by that, and we've written a review which came out in one of the journals fairly early on, putting GM-CSF up there as one of the potential disease markers which might be modifiable by anti-GM-CSF treatment. But it wasn't a particular feature of RSV disease. And I should also point out that, that the systemic changes in this model are relatively mild. So RSV is much more of a mucosal disease. It really is in the respiratory ciliated epithelium, and the systemic changes are pretty modest. I mean, we have found some peripheral changes, but by comparison with influenza, in which we're also doing human challenge studies, the H1N1 and H3N2. So we don't get as much of a peripheral change as you do with influenza.
I understand. So Valentina Eberlein has a very technical question. She thanks you for your interesting talk and asks a question about your PCR analysis. She asked, I was wondering why you have more L gene copies on day 0.75 than on day 4. Do you have an explanation for that?
Okay, so
Peter, can you show the specific slide?
Okay, yeah, let's go back. So we're talking about the slide in which we can see the. Oh, this is the viral load. So back at the beginning, right in the start of the incubation phase, if we take a sample the morning after we've inoculated the virus, you can still find some L gene, which we think is just a hangover from the inoculation the previous night. And we see that either in those with colds or without colds equally. But and then the level of the L gene declines, because from then on, you're only seeing de novo produced viral RNA. So that initial early detection is just a residuum of what's been inoculated. I think that's the right answer.
Yeah.
Yeah.
I'm not marking your answer, but it sounds like the right answer.
Thank you.
So I left a bit of a holistic question till the end. Maybe more come in, but this is the last question in the chat by Alireza Tahamatan, who's asking, regarding susceptibility and disease severity, how do viral host and environmental factors interact each other to determine the RSV disease outcome? I guess that's the million-dollar question, but maybe you can allude to that.
Yeah. Well, it's really the subject of a lifelong study, isn't it? And I think with every different viral pathogen, and indeed with every bacterial pathogen, you know, this interaction between the host and the microbe is the key question of interest that all of us need to focus on. You know, it's a multi-layered thing. There are clearly genetic determinants which are being elucidated for each virus, which are different in terms of what determines susceptibility. So in COVID, we just had a really lovely outcome from a big national study that we've been doing in the UK, identifying 3 novel gene loci which are variable in our population here, which determine the susceptibility to COVID. With influenza, we published a study showing the Mosaic study, which showed that there's variability in IFIT-M3, which contributes to severity. And with RSV, we ourselves haven't done genetic studies, but I think there have been genetic studies, particularly in the Netherlands, actually, which have shown an association with innate variability in innate genes, mostly, I understand. That hasn't been a particular focus for us, but it's clearly important. And then there's in addition to the direct genetic effects, there's epigenetic effects, and there's particularly the history of the mucosa in terms of what else has caused inflammation, whether it's a tendency towards asthma and atopy, or whether it's intercurrent or pre-existing infection. I think this whole landscape needs to be taken into account in order to explain the outcome. And trying to integrate all of this knowledge is very much a sort of lifelong area of study.
You're not done yet. That's an absolute conclusion.
So just to jump in for a sec, you mentioned these genetic studies. You probably remember Jeremy Hall from the UK, who did a fantastic, one of the largest genetic studies on IL-8 polymorphisms in relation to RSV severity. I think I don't think it has been forgotten, but people don't talk about that study much more. But in the light of your study, I think this is a very interesting finding that IL-8 polymorphisms define disease severity.
Yes, and also Paul McNamara and Ros Smith, when she was in Liverpool, they had a paper in the Lancet which was identifying IL-9 production from neutrophils as being an important factor as well.
Yeah. So I think somebody in the question box that we haven't touched upon is: do you think that neutrophils may be pre-activated by RSV from previous years, or reminiscent RSV? And in the same idea, do you think that maybe RSV-specific antibodies might be related, so pre-existing RSV antibodies might be related to neutrophil activation upon challenge? So I desperately try to bring back antibodies to the picture. You can see that.
Yeah. Well, I think that's something we need to discuss with our neutrophil biologists. I mean, it is perfectly plausible, isn't it, that antibodies could form a bridge and could cause some activation. And that I think goes back to the stories that have been around for many, many decades about the potential of antibody to cause some not only protection but some disease augmentation. I think it's an interesting area that certainly could be explored if these studies could be extended. Yeah.
There was this recent Science paper that I'm sure you read, that glycosylation, or the lack of fucose on antibodies against SARS-CoV-2, define disease severity in COVID. Just a steric hindering of interaction between the antibody and the Fc receptor actually define the signal. So conceptually, I thought there would not be a reason that would not be true for RSV, actually. Did you have a chance to see that?
Yeah, that's very interesting. Yeah.
Okay.
I think 2 more questions came up that are not answered yet. The first one is from Ron Dagan, who asked whether you treated your experimental subjects with antibiotics. I think the answer is no, I guess, but maybe you can allude to whether you are planning on to do so to rule out the bacterial impact on the pathogenesis through potential stimulation of neutrophils.
Yeah, I think we would only explore that sort of possibility if there was some positive indication that there was the presence of some bacteria that was causing this, and I don't think we have that at the moment.
Yeah. And then Adrian Wildfire is asking, is there a basic connection between CXCL10 and IL-10, as both seem to be raised and prognostic in both SARS-2 and RSV relating to mechanical ventilation?
Yeah, that's an interesting question. I mean, CXCL10 is often associated more with interferon activation. I mean, IL-10 is often present as a marker. I mean, it was classically an inhibitory mediator, but it often seems to be present in inflammatory conditions as well. And when we've looked over time, we haven't seen any different time appearance of that apparent anti-inflammatory and pro-inflammatory or reflective of inflammatory mediators. They don't seem to be sequential.
Okay. And then Sophia Buonocore comments, I think alludes to what we also was saying, that maybe the antibody quality or the and the FC-mediated mechanisms could be linked. And then she comments that maybe we can learn from the rescue study using system serology to link antibody quality and RSV disease severity. So I guess that's a promising work to come.
Very interesting suggestion, yeah.
And then I guess the questions raised by the people online are all answered, and I think it was a very lively discussion. Clearly, your presentation evoked a lot of thoughts, and I would like to thank you for this beautiful presentation, and I look forward to more data coming from this type of experimental setup. And with that, I would like to give the word back to Louis.
Thank you, Linde. You were a fantastic co-host. Actually, I didn't have to do anything, so thank you very much. Peter, you were very busy talking and responding to questions, but you didn't see how much applause you got from a lot of people that you know very well. People are really excited about your presentation. Personally, I thought, and I'd love to hear some feedback later on offline, that there was a good balance between the duration of your presentation and the Q&A. We had a very, very lively discussion. So I enjoyed that particularly. I think a lot to learn from neutrophil biology in RSV pathogenesis for the coming decade. So that's really reassuring that we're not going to retire yet. So thank you very much. On behalf of the ReSVINET Foundation, on behalf of everybody in the audience. With that, I would like to remind you that in March 26, I think, you will get a reminder, and we're going to talk about the impact of COVID on RSV and other viruses epidemiology in the southern and the northern hemisphere. I hope to see you again. Please fill out the feedback evaluation forms. They are very meaningful to us. And thank you, Leila and Sophie, for making this all possible. Have a great day. Bye.
Thank you very much.